So hello and welcome everyone to a webinar from TissueQuest. My name is Johanna Strobl and I work at the Medical University of Vienna. Today I would like to show you our data on long-term skin-resident memory T cells that proliferate in situ and are involved in human graft-versus-host disease. This paper was published in Science Translational Medicine recently, and if you want to check it out, you can scan this QR code right here. So first, a few words about graft-versus-host disease. Graft-versus-host disease is the major cause of mortality after stem cell transplantation, and it's commonly described as the destruction of host tissues by invading donor cells. Acute graft-versus-host disease is mainly characterized by a T cell infiltrate, shown here in the example of the skin, while chronic graft-versus-host disease is a mixed B and T cell infiltrate in tissues. Interestingly, when you look at the distribution of graft-versus-host disease in the human body, you can see that GVHD primarily affects organs that are populated by tissue-resident memory T cells, for example, the skin or the gastrointestinal tract. So we wondered whether tissue-resident memory T cells can survive the radiochemotherapy that precedes transplantation. So blood T cells are commonly eliminated by this radiochemotherapy and then replaced by donor peripheral blood stem cells. And for tissues such as the skin, this has not been investigated so far. So we sampled patients shortly before transplantation and took blood and skin samples and then looked at the distribution of T cell markers, for example, using TissueQuest software. And indeed, we found that while the blood T cells were almost completely eliminated, in skin we saw around 60% of T cells from baseline values. And also after transplantation, donor and host T cells coexisted in the skin and had a distinct transcriptional signature. And with this, we were able to identify potential novel markers for tissue-resident memory T cells. For example, we saw that the marker RUNX3 and also the gene galectin-3 were continuously upregulated in skin T cells. And also when we confirmed this on a protein level using immunofluorescent stainings, we saw that those markers could be potential novel markers for long-term tissue-resident memory T cells. So how long can TRM survive without replenishment from the bone marrow? For this, together with the team from TissueQuest, we developed a method to detect chimerism using StrataQuest software. And there we looked specifically at patients that were transplanted with peripheral blood stem cells from a different sex. So, for example, a female donor and male host, or vice versa. We then performed the immunostaining for different tissue-resident memory T cell markers, and this was followed by an XY fluorescence in situ hybridization to visualize the X and Y chromosomes within the nucleus. We could then overlay those two images in that we could detect the same cell in the immunostaining and in the fluorescence in situ hybridization. And this is the image that is shown in the StrataQuest analysis software, where we can see cells that express, for example, CD3 surface marker and then detect an X and a Y chromosome. And using this, we could see that while the blood T cells were almost completely replaced by donor T cells after transplantation, in skin, we had astonishing amounts of around 50% of T cells that were still of host origin in the skin up to 10 years after transplantation. So we next asked how TRM can survive in the skin. And for this, we first looked at T-cell receptor clones. And also here we saw that blood T-cell clones were completely replaced by the infusion of donor cells. And in skin, we saw that a large amount of clones was still shared between different time points after transplantation. So we also performed T-cell receptor stimulation of blood and skin T-cells on the day of transplantation, so when only the radiochemotherapy-resistant T-cells are still present. And indeed, while the blood T-cells were not able to proliferate after radiochemotherapy, in skin there was still proliferation that was significantly different from healthy skin T-cells. Those radio-resistant T cells also expressed the proliferation marker Ki67 to a certain percentage. And when we again tracked different T cell receptor clones, we could see that some clones that were present at baseline samples then even expanded upon radiochemotherapy and then remained as expanded clones in the skin after transplantation. So what could now be the impact of TRM on graft-versus-host disease? So we first looked at TRM presence during radiochemotherapy before graft-versus-host disease developed. And indeed, in the patients that later developed graft-versus-host disease, we could already, at the time of transplantation, see increased amounts of tissue-resident memory T cells. In graft-versus-host disease, tissue-resident memory T cells then showed superior proliferation, so they expressed Ki67 to a higher percentage than the other infiltrating T cells. We then employed the same technique I showed before with sex-matched stem cell transplantation and looked at recipient T cells in GVHD skin lesions. And here we saw huge patient variability, with some patients having up to 100% of donor host-derived T cell infiltrate, and other patients displaying more of a mixed donor-host infiltrate. So we investigated whether host and donor T cells can equally produce cytokines in graft-versus-host disease. And indeed, there doesn't seem to be an impairment of the host T cells in the ability to produce perforin, coenzyme B, or interferon gamma. Next, we looked at regulatory T cells, and surprisingly, the origin of regulatory T cells in graft-versus-host disease is mainly a donor origin, whereas only a very small amount of regulatory T cells are host-derived. So overall, I'm already at my summary. We could show that in healthy skin, tissue-resident memory T cells inhabit tissue and coexist with tissue-patrolling T cells. Those tissue-patrolling T cells are then eliminated by radiochemotherapy. Tissue-resident T cell clones survive, express TRM markers such as CD69, RANX3, or galactin-3. They can expand locally and clonally, and then contribute to graft-versus-host disease in that they undergo local proliferation and produce cytotoxic granules and cytokines and are then augmented by the influx of donor T cells. So with this, I would like to thank everyone involved in the study, also the team of TissueGnostics, Robert Nika, who helped us to develop this StrataQuest method, and my lab led by Georg Stari at the Medical University of Vienna. Thank you for your attention.